continue with the biochemistry flashcards. So here we have the Michaelis-Menten equation on a graph. So the equation is that the initial velocity of the reaction we're talking about here, I believe. So V equals Vmax times the substrate concentration over the Michaelis constant, Km, which is the concentration of the substrate at half of Vmax, plus the substrate concentration. So plot V naught found from the linear steady state parts of product versus time graphs with different substrate concentrations. Vmax is the fastest that a lot of copies of an enzyme working together can work. It does depend on enzyme concentration. If you want to know how fast one copy of an enzyme can work, divide Vmax by enzyme concentration to get kcat at high substrate concentration. Enzyme is the limiting factor. It gets saturated, so you get a plateau at Vmax. So as you can see here, we have initial rate of product formation, V naught, and then at high substrate concentration, enzyme is the limiting factor. It gets saturated, so you get a plateau at Vmax. Then at low substrate concentration, substrate is the limiting factor. Km is the concentration of substrate that gets you to half of Vmax. Km tells you about the affinity of the enzyme for the substrate, and Vmax tells you about the speed, right? And the Km does not depend on enzyme concentration. So regardless of how many enzymes you add to the equation, the affinity of the substrate to the enzyme stays the same. What type of enzyme catalyzes interconversion of isomers? That will be the isomerase, and an example would be phosphoglucose isomerase, which turns G6P into F6P. What occurs when two cysteine molecules are oxidized and create a covalent bond between their ethyl groups? This is a disulfide bond. What adds a phosphate group? We know it's a phosphorylase from an inorganic phosphate like HPO4 to a substrate. Phosphorylase. Glycogen phosphorylase adds a phosphate group to glycogen and just elongates the glycogen, right? What is the guanidinium group? It's the arginine, right? So Argentina versus Guantanamo Bay, they're close. What are used by the immune system? What are used by the immune system to target a specific antigen, which may be a protein on the surface of a pathogen or a toxin? These are antibodies, also known as immunoglobulins. What catalyzes cleavage with the addition of hydrogen? That's a hydrolase. And an example of a hydrolase would be like aminopeptidase, for example, that hydrolyzes the peptide bonds. An example here we had which I definitely forgot, is phosphatase. What pushes hydrophobic R groups to the interior of a protein, which increases or decreases entropy of the surrounding water molecules? That would be, so when you push the hydrophobic R groups to the interior of a protein, that would uh, decrease the entropy. Oh, the surrounding molecules entropy would increase, I believe. Yes, and it's the hydrophobic effect that does this. When multiple hydrophobic molecules are in water, the molecules group together spontaneously. The combined molecules have less surface area touching water, so fewer water molecules are needed to interact with the hydrophobic surface. This means more water molecules can roam freely, which is an increase in entropy, while for the molecule itself there is less entropy since it becomes an ordered structure. The hydrophobic effect. Cooperative enzymes display a what curve? A not hyperbolic but a sigmoidal curve. Sigmoidal because of the change in activity following initial substrate binding. So it's like a positive cooperativity where you get cooperative binding. The more molecules they attach, the more affinity it has. It exponentially adds so the tissue during exercise, and then the tissue at rest, and the lungs. So this is the oxygen pressure and the saturation of hemoglobins with percentage of the oxygen they're saturated with. The isoelectric point formula when there is a basic side chain is, so when it's basic, we know the carboxylic acid is pKa1, pKa2 is the amino group, so it's pKa2 plus pKa3 divided by 2. That gives us the pi for a basic side chain. Titration curves of 20 amino acids. Let's look at the basic ones. We can see that this is the midpoint, or this is the midpoint, and this is the equivalence points. It has two midpoints, equivalence points. So what is when the binding of the first molecule of B to A changes the binding affinity of the second B molecule, making it more or less likely to bind? This is the cooperativity, right? Cooperative binding results in a sigmoidal curve. This amino acid is glycine, GLY, G. Oh, sorry, shit. I thought yeah, I should have noticed here. Yeah, this is alanine, ALA, nonpolar aliphatic. What part of enzyme is the site of catalysis? It's the active site. This is the thiol group, indole group. So indole is like W. You can see it in the indole, which is tryptophan. Found in tryptophan. It's a phenyl with a 5 atom ring, one of which is a nitrogen. What theory states that the enzyme and substrate are exactly complementary and fit together like a key into a lock? This is the lock and key model. This amino acid is proline. It's when the functional group reattaches to the amino group, so it comes one C here, one C here, next C here, and the uh, third one is, I believe, yes, one, two, three. Third one is attached back to connected back to the nitrogen in the amino group. It's cyclic aliphatic, not aromatic, and it's hydrophobic, typically found in beta pleated sheets. It turns kinky, disrupts alpha helixes too. Proline shares many properties with the aliphatic group. Proline is formally not an amino acid but an amino acid. Interesting. It's imine, not amine. So let's flag it because it's a cool amino acid we want to have our eyes on. What theory states that the enzyme and substrate undergo conformational changes in order to interact fully. This should not be key lock and key model, but conformational model. Induced fit theory. Yeah, I forgot the name. Induced fit. Substrate entering active site. Enzyme complex. Enzyme substrate complex. ES. Changes shape slightly as substrate binds. And you leave the active site with different shapes. Y amino acids tryptophan, W, TRP, nonpolar. It's the 6-ring phenyl group attached to the 5-ring nitrogen on the side. And then the second carbon attached to the nitrogen is attached to the carbon, which is attached to the backbone amino acid. It's aromatic. Indole group emits fluorescent light. And if you remember which one absorbed UV light, it was phenylalanine, right? So you can flag this one too. And it's hydrophobic. What is a metal cation that is required by some enzymes? A metal cation that is required by some enzymes. That would be hmm. So let's look at metal cations that we have. Na plus, for example. Um, K plus, magnesium, calcium.
Oh, so we're talking about cofactors and coenzymes. So cofactors are inorganic, like metal ion, metal cation, right? Metal ions, coenzymes are subtype of cofactors, organic cofactor. And they have prosthetic groups and co-substrates. So the co-substrates are transiently associated, and the prosthetic groups are permanent. So prosthetic permanent. Prosthetic limbs are permanent limbs. So yeah, non-protein cofactor. Cool. What are rippled strands that can be parallel or anti-parallel beta pleated sheets, and they are a common secondary protein structure. And the hydrogen bonding and proline can introduce kink inside it. Rippled strands. What is an organic molecule that is required by some enzymes? This would be coenzyme type of cofactor. It's an organic cofactor. This amino acid is leucine. It has the L, and isoleucine is like this instead. So it's an L. It's hydrophobic. What inhibition of an enzyme is when an enzyme is inhibited by high levels of a product from later in the same pathway? So if too much product is, this is like a, we have positive feedbacks and negative feedbacks. This is like a negative feedback, but we're talking about enzymes here. So we have competitive, where the substrate is competing with the inhibitor for the active side, and the more inhibitors that sit on that, there's no more room for the substrates. So it's like a car that's going to the destination but is not bringing the substrates as passengers, and it's like fake passengers that's sitting in, and when it comes back, they're sitting in again. So these inhibitors sit on the active side, they're on the car seat, right? And high levels of um, so their inhibition, basically they're inhibited by high levels of a product from later in the same pathway. Is this competitive or is this non-competitive or uncompetitive? Now, the difference between on and non is that you have on read messages, your KM stuff is reduced, right? For, so is this related to Vmax or KM? So affinity of the substrate to the enzyme is about the km, but Vmax is the speed, right? I know we're talking about km here since the levels of the product does affect the enzyme activity. So I believe this is competitive. So this is all about feedback inhibition that I just talked about earlier. So this is like a negative feedback, right? So yeah. The isoelectric point formula for when a neutral side chain is pK1 plus pK2 over divided by 2. What inhibitor binds at the attractive side, active side, and thus prevents the substrate from binding? This is a competitive inhibitor. Prevents the substrate from binding. Can be overcome by adding more substrate, because more of the substrates would be competing more with those inhibitors. So your Vmax does not change, but your Km increases. So an increase in Km means that your curve is going to shift to the right, which means you need more of the substrate to fulfill half of the Vmax. So yeah, your Vmax does not change, however. Here we have a competitive inhibition where the substrate wants to sit on the active side, but the competitive inhibitor is already there. We have irreversible inhibition where the substrate wants to sit, but irreversible inhibitor is has filled the spot, active side. The conformational change of enzyme happens with the uncompetitive inhibitor. This is the second type. So the substrate is bound, but the conformation of the enzyme complex has changed. In mixed inhibition, if you have mixed inhibitor, the conformational change of the enzyme happens and the substrate binds or not binds. Then we have non-competitive, where there is conformational change of enzyme, substrate bound or not bound. Irreversible and reversible inhibition are broad categories that include the four major types. For example, competitive inhibitor can be reversible or irreversible. For this, we have the binding side for competitive is on the active side, the enzyme. For uncompetitive, it's the ES complex. And for non-competitive, is both ES complex or enzyme. It has binding of substrate. It's only for competitive. For the rest, it's not. It does not inhibit the binding of substrate. Effect on Km for the competitive, it increases it. For the third one, the non-competitive, the Km does not change. And for uncompetitive, both Km and Vmax decrease. Now for competitive, the Vmax does not change, and the Vmax for non-competitive decreases. Cool. I went through this a lot in detail. Like on the 1st of January, it was a month ago. So you guys should take a look at my video from the very first videos where I was basically reviewing all my quick notes. So yeah, this amino acid is lysine. LYSK is polar positively charged aliphatic basic, and I believe it is 2 or 3 carbons and then a nitrogen at the end. It should be 4 carbons and then an NH3 plus at the end. Since I didn't get it correctly, we can do this again. What inhibitor binds only with the enzyme substrate complex? This is the uncompetitive inhibitor. Nice. What is this? This is not leucine, or is it isoleucine? Should be. It might be isoleucine. Yeah, ILEI, nonpolar aliphatic. Let's do it in 6 days. Let's also flag that. What inhibitor binds at the allosteric side away from the active side? So we have allosteric side. It's the other one from the active side. So the one that attaches to the allosteric side is the uncompetitive inhibitor. Oops, non-competitive. It does not prevent the substrate from binding to the active side. Yeah, apparently the uncompetitive is yes competitive. Yeah, made a mistake here, so we do that again. Valine is the V, and then so it's three carbons. Yes, this is valine. Cool. In competitive inhibition, we have Vmax not changing and Km increases, goes up. Nice. The amino acid here is, so this is, oh, interesting. This should be glutamine, GLN-Q, polar and aliphatic. So arginine is R, but asparagine is N. And as asparagine is synthesized from aspartic acid and glutamine. So we have glutamine here and aspartic acid, you get asparagine. Asparagine is the shorter form with one less carbon, and you have the nitrogen. So we can flag this one too. In uncompetitive inhibition, we have the Vmax and Km both reducing on red messages. So it goes down. The isoelectric point when there is no side chain is should be equal to pH, or it's just like pK1 plus pK2 divided by 2 when there's no side chain, and it's H. The Michaelis-Menten equation is V equals V max times S over Km plus S. Yeah, this one is hard. Proteins is the local folding of neighboring amino acids, including alpha helices. And so local folding is tertiary structure. Oh, secondary structure of proteins is the local folding of neighboring amino acids, including. So I know why I got this wrong. In non-competitive, it's the Vmax going up, going down, and the Km not changing. Why? Because it's attaching to the allosteric side, and doesn't matter how many substrates you add. At the end of the day, it's all just not gonna stick. It's like the seats have changed in the car. The passengers cannot sit, and the Vmax goes down because you're just taking less passengers. This is also the Lion-Weaver-Burke plot is also important. So for the competitive, we want we want the lines to meet at the y-intercept because the v max is not changing, and for the non-competitive we want the km to not change, so the x-axis, which is 1 over s, and because it's reverse, um, we want both of them reducing, but it goes up, the inhibitor line goes up instead of down because it's reducing. And this plot, the um, Michaelis Merkin, the Michaelis plot is that's the inhibitor lines here are actually going lower. The normal enzymes like this, the competitive inhibitor is same Vmax, but different Km, lower Km. For the non-competitive, it goes uh, half of Vmax and the same Km. And how would the uncompetitive be? Both is reduced, so it's here, and also, so it's not only less, but it just goes forward, shifts to the right. Or your Km reduces, should shift to the right. Or it shifts to the left here. Anyway, amino acids are simple organic compounds containing both an amino group and a carboxylic acid group, carboxyl group, amino carboxyl. Lineweaver-Burke plot. The x-intercept is 1 over s, the y-intercept is 1 over v. The ratio indicated by the slope is so 1, so it's always y over x, right? So 1 over v over that, the other one is km over v. Km over v max, nice. The intercept is minus 1 over km. Okay. The amino acid is, this should be it's threonine actually, THRT, non-polar aliphatic. Nice. This graph shows activity of, so the 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 Vmax is um unchanged, so we immediately know that it is a competitive inhibitor, and the Km is increased because this is lower. What catalyzes cleavage with addition of hydrolase? And the example would be phosphatase. Nice, finally. This graph shows the activity of what inhibitor? Both Vmax and Km have decreased because they're higher, so it's uncompetitive inhibitor. The N-terminus of a peptide chain is positively charged due to NH3 plus. This shows, so higher V1 over Vmax means lower Vmax. Vmax has decreased and Km is constant, so it's non-competitive inhibitor. Secondary structure is stabilized by hydrogen bonds. Nice. Lineweaver plots are described as double reciprocal plots because the x-intercept is what and the y-intercept is what. Both of them reciprocals. Double x-intercept is -1 over km, and y-intercept is 1 over Vmax. Nice. Double reciprocals. This is arginine, ARG, and R, and it's positively charged polar aliphatic basic. And this is the guanidinium group. Then we have an irreversible inhibitor, which is any inhibitor that is what? To the active site of some enzyme, thus eliminating its activity. That blocks, covalently binds to the active site. Covalently. So this is we're talking about covalent bonds, like acid-base stuff like that. They're sharing not ionic bonds or hydrogen bonds or yeah. The inhibitor binds to the enzyme irreversibly through formation of a covalent bond to the enzyme, permanently inactivating the enzyme. Nice. What structure of proteins is the linear sequence of amino acids? Is the primary structure stabilized by peptide bonds? What inhibition is an irreversible form of enzyme inhibition that occurs when an enzyme binds a substrate analog and forms an irreversible complex? Substrate analog and forms an irreversible complex. The ES complex is this uncompetitive suicide inhibition. Wow. A special class of irreversible inhibitor. Inhibitor is structural analog to the substrate on which the enzyme act giving product. The products bind irreversibly with the enzyme. The product inactivate the enzyme. I didn't know that. Proline has a what structure? Rigid cyclic structure and can interrupt secondary protein structures. Rigid cyclic. Nice. Nice. What effector binds at the allosteric site and induces a change in the conformation of the enzyme so the cut substrate can no longer bind to the active site? This should be the non-competitive allosteric effector. Non-competitive in this, right? Positive effectors, activity goes up, and negative effectors, activity goes down. Structure of proteins compose the these are actin, myosin, and so they compose actin, elastin, tubulin, collagen, keratin, cytoskeleton, anchoring proteins, and much of the extracellular matrix. Anchoring proteins, like these cross bridges for muscle, myosin filament. And then we have cytoskeleton inside the cell, and extracellular matrix, even outside the cell. What effector is an allosteric regulator that is also a substrate? Homotropic. O2 is a homotropic allosteric regulator of hemoglobin. Wow. Homotropic allosteric enzymes. Substrate equals effector. Multi-subunit enzymes, the same binding site on each subunit functions as both active site and regulatory site. Substrate acts as an activator as well. This is why we have the positive cooperativity. Binding of one substrate alters the enzyme's conformation, enhances the binding of subsequent substrates. Sigmoidal kinetics, sensitive to small change. Heterotropic allosteric enzymes. Substrate does not equal effector. Homotropic. Motor proteins convert chemical energy into mechanical work by ATP, expanding hydrolysis of ATP. Hydrolysis, why? So because we use phosphatase to hydrolyze the ATP. We take one phosphate from it to get energy. Muscle contraction, vesicle movement, cell motility. Examples include myosin, kinesin, dynein. These are motor proteins. Nice. Blood effector is an allosteric regulator molecule that is different from the substrate. That should be a heterotropic effector. What pushes hydrophobic R groups to the interior of a protein? Its hydrophobic effect, which increases the entropy of the surrounding water molecules. Nice. What is the chemical addition of a phosphoryl group, PO3 minus, to an organic molecule? The addition of phosphorylation. Yes. Phosphorylation, PO3 minus is added. Kinases do that, right? ATP, add phosphate. Kinase brings ATP to do that, right? Then the phosphorylation target, it gets phosphorylated, and water, and phosphatase uses water to get that turned out phosphorylated molecule, protein for example, into an unphosphorylated one, and then here you can see the transferred phosphate group. Good for now. What theory states that the enzyme and substrate undergo conformational changes in order to interact fully? This is the induced fit theory. What is the chemical addition of a carbohydrate? Carboxylation, glycosylation. Glycosidic bond here between nitrogen and carbon. You are adding a carbohydrate. So glycosylation is what it's called, glycosidic linkage. We also have for sugars. Asparagine can be N-linked, serine threonine can be O-linked. The enzymes responsible for glycosylation don't recognize anything else. Can glutamine be a substitute for asparagine? Not for glycosylation. Why not? Both have amide for their side chains, and the only difference is an extra carbon. The enzymes responsible for glycosylation don't recognize glutamine. What's specific about it? It's an enzyme, so probably the size shape of the active site. Glutamine doesn't. Toddler needs sugar. STN. Toddler needs sugar. What is metal cation that is required by some enzymes? Cofactor. Precursors to an enzyme. Substrate. Zymogens are precursors to enzymes. Finally, this is what zymogens are. We learned that we have deuterions that are the neutral form of an like an acid base. Yeah, so that's when the pi equals isoelectric point equals to pH. The compound is in its deuterion form, the amino acid, for example, right? And then zymogens are precursors to enzymes. We have environmental factors for the zymogen, and then autocatalysis activated, and then we have the active site. Cool. Exergonic reactions require or release energy.
So exergonic reactions release energy, right? Michaelis-Menten reaction scheme is where you have the normal enzyme and then you have the so the one that the Vmax is unchanged for is competitive, so it's the same, but it's just like a steeper. I mean, the Km is different. Oh, so we're talking about the reaction. It's E plus S, the enzyme plus substrate, and then you have ES, and then enzyme plus product. K1, the binding of the enzyme to the substrate, forming the enzyme substrate complex. The catalytic rate, K2, the catalysis reaction, producing the final reaction product and regenerating the free enzyme. This is the rate limiting step because it's irreversible, right? Endergonic reactions require energy. These are not spontaneous. Endergonic, you're going up, the delta G is plus, but here delta G is minus in exergonic, so energy is released. What does this look like to you? If it looks like a plain head, you're an adult. If it looks like an elephant being eaten by a boa constrictor, you're still a child at heart. If it looks like a free energy reaction diagram, you're in organic chemistry and your life is over. Why so dark? What protein is any protein that acts as an agent to bind two or more molecules together? Binding protein, DNA binding protein, single strand binding protein, telomere binding protein. What are complex proteins such as hemoglobin consisting of amino acids combined with other substances? Complex proteins like hemoglobin consisting of amino acids combined with other substances. Um, Conjugated proteins. So finally, these are what. So an example of conjugated protein is hemoglobin. These amino acids combined with other substances like heme. Prosthetic group is the heme, the non-protein part of a protein. Required the heme molecule is the prosthetic group. Let me take a break and we'll be back soon. Peace.